In today's A-level IB video, we're going to look at absorption and specifically within the small intestine. So how do various substances, so soluble food products, pass across the wall of the small intestine into the blood? And that's what today's video is all about. So absorption in the small intestine. So there are four main methods. The first one is the one you'll be most likely to be familiar with, which is simple diffusion. Next up, we have facilitated diffusion. Number 3 is active transport. And then number 4 is endocytosis. So we'll go through each of these methods of transport in turn, giving specific examples. So absorption in the small intestine. We know that we're passing small soluble food molecules move into the blood. So firstly, simple diffusion. We're going to list first of all some nutrients which move in this way. This includes fatty acids and monoglycerides. Notice that in terms of their properties, these are water-hating, they are hydrophobic substances. So just to add a few extra notes, simple diffusion, remember, occurs down the concentration gradient from an area of high concentration to low concentration, and this is a passive process. No ATP is required. Next up, facilitated diffusion. This is for the hydrophilic substances, so water-loving substances. Our key example here is the sugar fructose. Remember that in facilitated diffusion, again, we're moving down the concentration gradient from an area of high concentration to low concentration. So far the same as our original point. It's a passive process, but importantly, you must notice that it requires specific channel proteins which exist in the plasma membrane. Next up, active transport. Remember that this is against the concentration gradient, so very different to diffusion. Therefore, we're going from an area of low concentration to an area of high concentration. This therefore requires energy from ATP, and it involves the use of specific pump proteins. Your key examples here are mineral ions, such as sodium, calcium, and iron. Last but not least, endocytosis. Remember, this is when we use vesicles to move substances into cells. Just to summarize, small vesicles enclose the digested nutrient, and your key example here is cholesterol, which is found in lipoprotein particles. And then crucially, we can't forget glucose. After all, glucose is the number one digested food product. This moves by active transport. The reason I didn't list it above is because it's quite a complicated process. It involves the use of sodium co-transfer proteins. And remember, these work by moving glucose together with a sodium ion across the membrane. So the glucose is moving against its concentration gradient, and then the sodium ion concentration gradient is maintained through its pumping out by a pump protein.